So this panel discussion is on the topic of CRO and lab automation, as mentioned, and is part of the AI in Biotech initiative by Nucleate. Nucleate are an organization that is student-run and global and wants to empower the next generation of biotech leaders and entrepreneurs. You can find more info about Nucleate, of course, on the web and on a link tree that the team will send in the chat shortly. On top of this, we're also putting together a CRO and lab automation resource page with all the resources that we'll mention today in this panel, and then more, both in terms of software, hardware, and then in general about outsourcing laboratory experimentation. Myself, I'm Lorenzo. I'm gonna chair the panel today. My background is in software engineering and neurotechnology. I run a software company myself, which is a hiring platform. So not related to biotech, but on the side, I'm very interested in all things biotech, in particular, of course, computational stuff, and also complex diseases modeling and age-related diseases in particular. And I'm also collaborating with the Nucleate team, and that's why I'm chairing this this panel today. On the panel, we have Simon from Bio, we have Staffan from ai.bio, and we have Alfredo from Briefly Bio. What we'll do now, we'll probably get started with just like maybe a round of introductions by all our panelists, amazing panelists today, and then we'll go a little bit more into detail of the topics of the day, so CROs and lab automation in particular, and then I think we can end up with some more general questions about, you know, the outlook of the industry, what's gonna, you know, happen in the future, what excites us, and then open up some questions also from the audience, which you can already put in the chat down there. So we'll go through the questions in the chat at the end. So I think without any further ado, we can get started with maybe the panelists today introducing themselves. I'd love to get maybe some background about yourselves and a little bit about the companies that you run and why you decided to start those companies, maybe. So background, company, and why. And maybe we can start from Simon if you want, then Staffan, then Alfredo.
Perfect. Thanks, Lorenzo. I'm Simon, co-founder and CEO of Vial. We started the company about 3.5 years ago, and our mission has been to reimagine clinical trials. Clinical trials are very inefficient today. They're still largely on paper. They're still largely in PDF and managed manually. And we've effectively built an end-to-end technology platform. Think web apps, think tablets, think backend integrations that run in a clinical trial in a highly digital way. And we now run that clinical platform both for others. And so we're running about a dozen trials for other biotech companies, as well as for our own internal drug discovery unit called ai.bio. We're based here in California. I'm in San Francisco, and we have about 85 employees.
Thank you.
Nice. Yeah. My name is Staffan, and I'm co-founder of Briefly Bio. My background is not in the biospace. I come from an engineering background. So back in 2018, I was still working for Volvo in Sweden, the automotive company, trying to usher in the self-driving revolution that's still not here. And then I figured there's this cool space out there called biotech. And so I decided to join a new company in London called Lab Genius. They're doing drug discovery, protein therapeutics. So I came in there, tasked with starting the automation function. And I was thinking, automation, easy peasy. Had to step into a lab for the first time in my life and kind of realized that this is a different game. You got to understand the lab work before you can automate the lab work. So it took me a while to really become productive, but after about 6 months or so, we had started building out the tech stack of wet lab automation. Also built out the team over the next 4 years or so at Lab Genius. So then we were up at 2022 or so, and so then I did some consulting in automation as well with a few different companies. But basically at Lab Genius, me and my two co-founders here at Briefly, we saw a lot of inefficiencies in how science is done in general, especially as it pertains to the documentation and the lack of love for the scientists themselves in the lab. And so we really we wanted to build something for the scientists to help with kind of the lack of reproducibility and the the symptoms of that in science. So we started briefly at SLAS, the big conference for automation and screening in Boston. We had the tagline, "This is not an ELN," which obviously means it's kind of an ELN. But the ELN comes with these, is boxed as a thing that comes in top down in a company. It lands on the scientist's head. Now they have more stuff to fill in. It's not really helping them. And so we want to change that. And so briefly, it's an experiment builder, and it's supposed to help scientists first and foremost. And as an effect of that, we get better documentation and more reproducible science for the organization. So yeah, that's me.
So I'm next. Yeah, I'm Alfredo. I'm the co-founder and CEO here at ai.bio, where we're building a cloud computing platform for biotech companies. Currently, what these companies do is they set up AWS, and then they have to spend millions of dollars in salaries and testing and building on top of AWS to configure it to then do like the transformations and the transformation, the storage, and the processing of high throughput data. We are building a cloud that comes pre-configured to do that, the workflow orchestrator, the file storage, the Jupyter notebooks, the databases. And by doing that, we save these companies all this time. Today, we have about 60 biopharma companies that are using us as their main cloud computing platform. We're a team of about 20 people. We've raised about $33 million. And yeah, hopefully we're just getting started. But thanks for having me today. It's very exciting to be along three such great individuals, and yeah, excited to talk a bit.
Absolutely, thank you guys. So I think we can get started on the topics of the day. Maybe we can start from the topic of contract research organizations, and for that, maybe I'll ask the first question to Simon, which is whose background is probably most relevant when it comes to this topic. And so for the ones in the audience that are not really familiar with the concept of CROs, what are CROs, and then why should anyone consider outsourcing the lab work to a CRO?
Yeah, I think CROs are a pretty widely used term, both for discovery, preclinical, and then clinical. I think that's probably the key set of distinctions. We focus on being a clinical stage CRO, and so the second you've prepared to file your IND with the FDA, that's really when we get involved with a biotech company. And so most companies at that stage come up with the decision: are we going to build an in-house clinical operations team and run clinical trials ourselves, or are we going to outsource to a CRO? And that's a pretty classic question. The trade-offs are usually some combination of expertise, scale, and cost that you can get outsourcing versus the challenge of having to hire a team that would only really be used for that one clinical trial with potentially less talented staff than you could get outsourced, and with the cost trade-off of doing it in-house. And so what you typically see is the vast majority of biotech companies do outsource, and they're outsourcing the scope of services that include project management, someone who oversees your clinical trial, data management, someone who reviews the data as it comes in, vendor management, biostats, powering your trial correctly, reg affairs, writing your protocol, writing your investigator's brochure, CRAs, contract research associates who are going to the clinics to ensure the data is high quality, reviewing all the documentation that comes in. I can keep going. It's a very large suite of services to run a clinical trial, and that's the scope that's usually looked to be outsourced.
Yeah, great overview. And I'm curious, what are some of the challenges that you see biotech firms that maybe come to you or other CROs have when it comes to actually delegating the work or part of the workflow to outside companies? Is there anything that comes to mind in particular?
Yeah, and everything's challenging. It's super expensive. It's incredibly slow. Picking a CRO is really difficult to do. And then that's just the first decision you have to make. You have to pick your vendors, you have to pick your sites. And so many companies think they've made all their decisions by the time they pick their CRO. But that's really just the first decision. You then have to say, who's going to be my central lab vendor? Who's going to be my EDC vendor? Who's going to be my ePro vendor? Who's going to be my vendor for all these different things? And the challenge is not just picking those vendors, but thinking how they're going to iterate and talk to each other. I like to make the analogy, it's like building a building, and your CROs, the GC, and then you have subs who come in. The challenge with building is that typically the subs are different for every project. There aren't systems to talk to each other. The project management tool doesn't talk to the billing tool doesn't talk to the analytics tool. It's exactly true of clinical trials, where the CRO usually works with a different set of different vendors for every trial. And so as a biotech company coming in, you want to be thoughtful about the choice of CRO. Also be very thoughtful about this decisions to support vendor. We haven't even talked about sites. The difference between choosing an academic medical center versus a private practice can mean a difference in a trial that starts in 6 months to 12 months, or a trial can start in 1 to 2 months. And so decisions on sites can be huge. And that's just the trial startup period. Thinking about enrollment, how many sites you need, that's a key decision factor that comes down to weighing pros and cons of different sites that are selected in a trial. So there's challenges galore that biotechs face in picking a CRO and then all the operational decisions that they're in.
Yeah, and just one last thing before moving to the other panelists as well, on this. I'm curious if you see any issues from maybe founders of smaller biotechs as well, giving up control of what they think it's something that they should control in terms of timelines, quality, as a sort of, you know, research process, to someone else who they can't really control the same way. Is that something that you actually see, and how do you manage that in terms of expectations?
Yeah, it's a huge trade-off that companies are making by outsourcing. This is not just a challenge at the small biotech company level, it's also a challenge at the big pharma company level. You can see from public statements that most of the big pharma companies outsource, and yet typically they're dissatisfied. Typically they're reassessing those decisions. There's many cases now of them attempting to bring clinical trials back in-house. But in order to do that, it's a tens of thousands of person organization built. So it's incredibly challenging for big pharma to do. And that same challenge, that same trade-off decision is not only happening in pharma, it's also happening at biotech. The only challenge at the biotech level is companies usually have 1 or 2 programs. And so the cost trade-off really rarely makes sense, where if you're hiring a team of project managers and contract research associates and a data manager and biostats, before you know, it makes no sense to have that team of people built out for only one clinical trial. So the cost trade-off just doesn't really typically make sense. But the time trade-off for a trial is really challenging. One of the challenges we see a lot with biotech companies is they'll go to a CRO, whether it's one of the big 4, big 5, kind of global multi phase large trial focused CRO, and they're bottom of the list. That big CRO makes 3/4 of their revenue from Pfizer, from GSK, from Roche. If you're the small biotech company with a phase 1, 2, you're not going to be anywhere near the top of their priority list. Or you can go to a small CRO that's maybe tailor-made just for you, more nimble, more agile, something like Vial, and hopefully you're going to have a better experience. So that's some of the considerations that go into the build versus buy and the cost and speed trade-offs.
Absolutely. No, thanks for the overview. I think we can move to the other topic, which is lab automation, for a second. And then we'll come back to CROs with some more questions, I think, later on, of course. In terms of lab automation, I think we can probably redirect most of the questions to Staffan, who I know has a lot to say on that particular topic. And then we can also ask some questions around Briefly Bio in particular. But in terms of lab automation, I think similarly to what we've done for CROs, could you define what we mean by lab automation usually, and what are the most common types of automations?
Yeah. Let me just first state, because when we talk about automation in a lab setting, we always go to the wet lab and like the robots and all that cool stuff. But there is the whole other side of things, right? When the results come out, all the automation that happens afterwards in software, right? Which is almost as big of a field. But yeah, so I come from wet lab automation, right? So defining what that is, I would say it is, for the most part, taking processes that a bench scientist have made to work with a pipette in hand, right? And now, for some reason, we want to either scale them up in terms of throughput, or we want to increase, consistency and standardization, or perhaps get some hands-off time by using robots. So we then need to take that, which was created for a human to do with a pipette. Sometimes we need to convert it, so we need to change the actual science, maybe get a different kit in. You can imagine something is made for centrifugation. Now we need to move over to magnetic bead handling instead, stuff like that. Get it ready and appropriate for the robots or the equipment we're going to use, and then program that equipment and then validate what we have in the end. Because just because the science worked when we started our automation journey doesn't mean that it's working when the robot is doing it right. Or maybe it's working, but it's not meeting our quality standards, stuff like that. So I don't know if that answers the question, but it's kind of a general view of what lab automation looks like.
Yeah, that makes total sense. And maybe for a second, let's focus first on hardware, so robotics, and then we can talk about software as well. In terms of implementing hardware solution, what are some of the considerations, some of the challenges that you see when it comes to implementing those things? I'm thinking about costs and all those factors to consider.
Yeah, good questions, big questions. I'll name a few topics, I think, and I think the first one actually has to do with the organization and people, which is like deciding what to automate for the organization. Who are we automating for? Who's going to use the automation we build? What is it going to do for the business? Like we can't just automate, in a vacuum because it's cool. So where does it fit into the business is one big question, and does it make sense for the business? The other one is the hardware, right? This is not just software. We're not just writing scripts. These are things that need to have a place in the lab. They cost a lot of money. They need to have their own consumables, like each liquid handling platform, for example, often have its own set of consumables, which needs then inventory and all of that. So that's one thing. And then further on is then the question of the actual science. Is the science ready for automation? Which is a big point of friction and problems that you try to automate too soon. For example, you have something that worked once on the bench, and now you move it on to robots, and then the next batch of samples fails, and now you have a way harder troubleshooting problem on your hands. So it's about maturity of the process. And then, as I mentioned before, also after you have developed some automation, you have big questions around how do you know that you're successful? How do you know that you're done with the automation project? But going back to cost, because that's usually something people think a lot about. Robots are expensive, but I think the capex is not the biggest problem around. Now we're talking about early biotech companies. If you're like a 10 people company or even smaller, like investing 50 or 100,000 in a robot when you've raised money, it's a lot of money, but it's not like that. That's not the big. That's not the big tension around pulling in automation into your company. The big thing is the time and the distraction it can become if it's not the right thing for your company. You can dedicate a big portion of your team to try to make automation work. And if you're not ready for that, that can become quite a big distraction, I would say.
Is there a particular stage that you think biotech companies should start to think about automating, or is something that you can do right from the start?
I think you can do it right from the start, but I think there should be a reason, kind of. We shouldn't just pull the automation card right away unless there's a reason for it. I think what's more important then is to pull a standardization card to say, okay, before we invest in robots, let's see if we can do this in a standardized fashion with humans. So basically, you think about the human as the thing executing what you would like to automate in the future. Because if you can't make that work, you're probably not ready for automation yet. That being said, though, I think there's other fussier values from starting to think about automation earlier, such as the culture. Like you need to build up this expectation that we're a company that does automation. We expect that our science should be robust enough to be able to run on robots and all that sort of thing, which is super hard to quantify. So yeah, in the end, the answer is not very clear. I think you can start thinking about it, but maybe try to take a few steps on the human route first, trying to get them to execute as a robot before you pull the trigger. It's some general advice, I guess.
Yeah, in some other words, you need to know what to automate before automating, exactly. If that makes sense.
I mean, I think so. There are other ways of doing automation. Like you can build walk-up automation where you kind of enable a very flexible type of automation. I don't think that's what most early stage companies will do because it requires work.
Yep. Absolutely. thank you for this. We'll come back to you with more question about Briefly Bio and how that helps, automating as well. But I would love to ask some questions to Alfredo, mainly around software, because of your background, I guess. And so I'm very curious about like, what are the software use cases in the lab? What are the most common use cases? And then in particularly for large bio, how does it help with those use cases?
Do you mean just software in general, or software within automation? Oh, you're muted.
Pardon. I was thinking mainly automation, given the topic of the talk. If you want to expand beyond that, it's fine as well.
Oh, totally. I think automation makes sense. I mean, when you think about automation on the software side, you're usually going to be thinking about kind of two sides, kind of the wet lab and automatically recording a lot of the stuff that is happening in the wet lab. And it's not just recording it, because maybe you're recording it, it's a little too advanced, but a lot of the times, like the formats that scientists like to fill out their information in is not the same like formats that is going to be computer readable in, say, a bioinformatics workflow. And so that also counts as automation, the ability to turn that into, and you'd be surprised the kind of stuff you see there of someone kind of filling out, like, so for example, just to be concrete, like scientists really like sheets. I mean, I love sheets. It's like super interactive. You can have multiple people changing values, but you can imagine that it's not a very like type safe interface to be filling out your values. And so the automation of turning, I mean, for an algorithm, for a scientist, if they kind of fill out a value as like, let's say like just capital P53, and then like the other one, they fill it out as like not capital P and then 53. Those for a computer are two different values. For a scientist, they look at it like, oh yeah, obviously it looks the same. And so the automation to go from that into something that's computer readable for the bioinformatics workflow, I think that's kind of where Briefly Bio fits in, and obviously doing some amazing work there. And that's a really hard problem. The second part is, okay, you have everything ready, all the results are ready, and you want to run a pipeline, right? You want to run a bioinformatics workflow, you want to do some analysis on the data, and the automation there is usually comes in the sense of bioinformatics pipelines. And you're gonna, like when you're doing a bioinformatics pipeline, say like RNA-seq, you're not doing like a single step, you're doing an array of steps. First, you're gonna have to like trim it, then align it, then you're gonna have to like do some counting, and then finally you're gonna have to come out with the final result. And so that's the kind of automation we deal with. And obviously, like I really can't like, okay, so obviously the big goal of automation is to just like you putting your data into the sequencer, let's say like an NGS machine from Illumina, and then the next thing you hear is an email in your inbox where you get back the results in a PDF, and it's just like analyze results. And that's awesome. And it sounds like, as a technologist, that sounds incredible. But that really is not better than you manually going through the analysis. It's not better or worse. It just happens to be at a point of maturity of your bioinformatics that makes a lot of sense. Now, when you're getting started, if you try and get started with that, you're going to run into a lot of trouble. Just like we've already emphasized in the lab automation, that's also going to happen in the computation. If you focus on the automation and making it all like, it's going to be very hard to debug. It's going to make it way harder to do everything. And so you should really start from a very pragmatic, like, let's do this all manually. And I actually, I even use this principle at our company when like people tell me like, oh yeah, I'm going to automate this thing. I'm like, no, like do it manually for like a few weeks. And then when it's like so clear that you have the right things, that you have the right values, that you've perfected and optimized it, then start automating it. And I think that's equally important on the wet lab as it is on the bioinformatics sense. And the platforms that we're building need to take that into account. And so we don't tell people like, oh yeah, put in your workflow, launch it into like scale. It's like, you should probably just start with a Jupyter notebook and just like do a bit of analysis there. When that Jupyter notebook is ready, then like run it a few times. And if you're not changing the values every time you run it, and you can truly have like one cell at the top where like you put in all your values and then every other cell just runs automatically, okay, you might be ready to create an actual batch pipeline that you can put into, in our case, Latch workflows, in other people's cases, AWS batch. And then at that point, then you put into that and you start debugging that process. And when that is working really well, okay, like now let's think about connecting it to the sequencing machine. Now let's think about like emailing the results back to the scientist without checking them manually. And so it's a step-by-step process, and you really don't want to get ahead of yourself. And there's really no better or worse. They're just different levels of maturity that you should be looking at.
Beautiful, beautiful. And I think you mentioned Google Sheets. And so my question here would be, what are the challenges to get, people who work in the lab, scientists who might not be necessarily used to the latest, software tools to actually adopt those tools, and in the interest of, speeding up, standardizing the processes. Is that something that you came across as a challenge? And I'm curious, how do you go about it?
Totally. I mean, we still come into that problem, and it's a problem we face every day. And we are trying to solve it for a few of the use cases. For other use cases, I think Stefan and Benchling and other solutions would be much better fit to do it. But for a few of the problems we come up with, the way we look at it is very pragmatically, similar to the way I see kind of synthetic meat and synthetic meat, synthetic proteins. It's like you cannot expect the user to do the thing just because it's better somewhere downstream. You need to build something that is currently better, that tastes better, that is a better user experience, that is like actually just makes their job easier. And I think Stefan touched on this a bit, but like that's how we look at it. It's like, okay, we want to like make it better than Google Sheets. Like we actually want to make their life easier. And how do we do that? And well, for us, that requires like really incredible software engineering, really incredible attention to detail, lots and lots of iterations, and doing that over and over with many customers until we have a product that people truly enjoy using more than Sheets. And you'll be, I think you'd be surprised how high of a bar Google Sheets or Excel is. It's actually like very mature products. And so that's what we're shooting towards. Yeah.
Makes a lot of sense. They're products that they've been along for ages, basically. I'm curious, Stefan, what's your take on this as well? What challenges do you see specifically, maybe with Briefly Bio, in terms of getting people to adopt some software that they might not be used to?
Yeah, I mean, I can just echo what was just said. Obviously, completely agree, and I think it's super important point. In the past, software was built for scientists and forced on them, and I think we're newer companies now. We're trying to change that a bit. It's still difficult, like Alfredo just said. We're up against very mature products that work really well for what they're doing, even if they're not optimal for the use case. But I think it comes down to just what he said. You build something that is useful for the person, not for their future self, not for their friend, not for their boss, right? But for them there and then, the problems they have, what they need to solve, what they need to do. That's what the product. That's where the product need to shine and be really good, right? And then you get all the other stuff, and that will be appreciated later. But we can't kind of sell in a product. Maybe we can, but I don't think it works, selling a product to someone and saying, hey, you get through the pains of using this product, and down the line you will reap the rewards. I don't think that works anymore. So yeah, it's a challenge, but I think if you do that, it's gonna, like, the product will start working for people, and then they will want to adopt it, right?
Yeah, absolutely. And still on this topic, I'm curious, then what is the main benefit, especially for something like Briefly Bio, in terms of maybe standardizing the protocols and the experiments, also thinking about, automation? How does that help with the whole workflow?
Yeah. So I can start by like, telling the story of me as an automation engineer. I'm tasked with automating things. I have to work with, one day a cell biologist, the next day a molecular biologist. They all write up their protocols and their experiments in completely different ways, because it's all free text, right? So, free text gives you the flexibility to write whatever you want, and that works really well with lab work, which is also very flexible. But it also creates this kind of a mess of the documentation, inconsistent formats, and all of that you, as an automation person in this case, have to then deal with. And so I think if we can, what we want to do at Briefly, right, is we want to build a tool that helps scientists do their work, things like plate layouts, calculations, and merging different methods together, stuff like that. and by having a consistent format that the output of this like a visual structure, we can all kind of have a similar understanding and more of a similar language around what does a protocol look like, what does an experiment look like. You can come in, you can be familiar with what it, the things that are there, and then we can start improving things like completeness of that protocol. And we have a common artifact to work on that's understandable. I think the reason why we struggle so much with plain text in automation is that we often want as much information as possible to be added in. We want the completeness is super important. But for someone who's following this protocol as an instruction set in the lab, more information is sometimes bad because it creates clutter in the protocol and it's not as clear anymore. So in free text, we get this tension between completeness and how clear and useful the protocol is. And that's what we're trying to change at Briefly, so that we try to take away that tension so that we can have fully complete protocols without that clutter, basically.
Awesome. That sounds great. I'd like to go back to the previous topics, just for a second, of CROs, and maybe go back to Simon again, just to finish up some questions around that topic. One thing that comes to mind is, let's say, I'm a early stage biotech founder, or even not early stage, later down, which might be the case for many people in the audience. How do I go about finding the right CROs for my need? Because what you guys focus on is clinical trials, but there might be other CROs focused on other steps of the research process. So how do I go about finding the right one for my needs, and how do I go about finding Vial if it's the one that I'd be looking for?
Yeah, great question. I think that the challenge with any procurement process is, how do you have sufficient breadth, and then how do you go deep enough that you ensure that you are working with the right partner. And so meeting, I think what I typically hear from biotech companies is they meet about 4 or 5, and they try and get diversity within that 4 or 5. So they'll go one very large company just to get a sense of what it could look like to work with a very large CRO. So they'll get a quote from one of the very large ones. They'll work with one that's therapeutic area focused, and so they kind of get what does it mean if I spike on expertise, and then go get a few that are kind of maybe more generalist but a good fit size wise. And so then you get a diversity of different types. And if you need to double click into one of those types and you really like the quote you got back from one of them, you can go deeper. But the world of CRO selection, at least for clinical stage companies, tends to be very formal. Tends to be a RFP process with at least a 3 or 6 month timeline to come to a decision. These are multi-million, if not $10 million plus decisions with a lot of different stakeholders, and typically are very very regimented in how they go about making that decision. So yeah, I would say diversity of CROs in breadth, and then go deeper where you find interest or kind of initial match with your needs.
Right, makes sense. So different CROs for different needs for different stages of your research process. You mentioned multi-million long timelines. That sounds very scary. How do I go about making sense of the pricing that maybe a CRO comes back to me with? Are there some benchmarks? And in terms of timelines as well, how do I make sense of the timeline as well? Does the CRO itself help with that, or do I need to rely on someone else?
Yeah, hard question. Really, the challenge comes down to having a really strong clinical operations leader internally. If I was a biotech founder, founder-led bio style founder, I think I would be looking to bring on a really strong clinical operations leader who could own those decisions. In many ways, the analogy would be for a SaaS founder thinking through GCP versus AWS versus any number of other hyperscalers. The question is, how do I trade off cost and speed and the extensibility of the platform? In this case, it's how do I think about also cost and speed, and how do I really kind of weigh those decisions? And that's a highly technical challenge. So I would look to hire a strong clinical operations leader who can help make that decision alongside the founding team.
Yeah, awesome. That makes sense. And in the same theme of thinking about things that could be relevant for the audience, I know there's a theme of people like me with a software engineering background that want to get into biotech. And maybe this question is again more relevant for Alfredo because of the of your background. As a software engineer or computational person that wants to get into the field, what are the challenges? Because I think that probably is something that you faced yourself. Correct me if wrong. What are the challenges of getting into the space, and what are some things you wish you knew when you did that yourself?
Interesting. So yeah, I mean, the field thankfully needs a lot of software engineers, and so that is the good part because it means that there is because there's so much work to be done. There's also a lot of ability to come into companies with the expertise in software engineering. You do that part, and meanwhile, they teach you about the biology that you're going to have to know. And when I say they, I really mean like you self-learning, and then they kind of expect you to not have that much biology, because if you can find an incredible software engineer who is also an incredible biologist, like they exist, they're just unicorns, right? And so as a biotech company where you're expecting to have complex software systems, you can't expect to have that. And so you need to expect people to be good at one of the two. And so thankfully, like we really need software. Thankfully, like these companies are hiring, and like having biology expertise is not a requirement. Now, I think partly, given that, you can, like, what I would recommend is either looking first at the truly just like software focused companies. That's really if you're an incredible software engineer, that's really going to be your best bet. If you want to continue being around incredible software engineers and being just focusing mainly on software engineering, you should go to one of the new age software for biotech companies. Obviously, ai.bio. We're always hiring software engineers. Please DM me if you know of anyone great, but also Briefly Bio, Benchling, and many other like really cool startups coming up. Vial is obviously another one of them on the more like CRO and clinical trial space, and lots of incredible companies. But that's kind of if you're a software engineer trying to get into space, and then you're gonna have to learn a bunch from there. Because, and this kind of merges with the topic of if you go in directly into the biotech, you need to be very pragmatic about software. Bio is not software. Biology is not the same. And we have one of the biggest challenges we face as an industry is we're a little bit allergic to software because we've had one too many tech bros come into the industry and be like, oh my God, I am your hero. I am here to save you. You're not using software. I'm going to make you software. I'm going to save you. And it really doesn't work that way. I mean, biologists, biotech people, they're incredibly smart. They're incredibly smart individuals. And they, believe me, they know about software. And if they're not using it, it is for particular reasons. It is for real blockers. It is for challenges. And so if you're a software engineer that goes to work at a biotech, or you're joining these companies and you want to truly contribute to one of the software and biotech companies, you're going to have to learn the bio, and you're going to have to learn enough to ask these biologists, why are you doing things the way that you're doing? Like, I believe you are a very smart person and you've made these like decisions. I don't even want to call them bad decisions. You've just made these decisions to use scripts, to not automate anything, to use Excel. Like, you've made them for a reason, and probably for a very good reason given your work. So tell me those, and maybe, just maybe, we can build tools that will help that be better. And we can iterate with you, with your constraints, to help those tools be better and to make better tools for you. And maybe we can't, because like software is not magic, and like your biology is the most important thing. And the biology at these biotech companies is what truly moves the bar, not how pretty their software is, how nice their UI is, or whether they spend. It's like the typical example is like, oh, I optimized your pipeline and I made it like from like 5 hours to now it takes 10 minutes. And it's like, dude, like my cell culturing takes 7 days. Like, I don't care if like the extra 4 hours that I saved there. And so just being very pragmatic and not being like that kind of tech bro mentality, and just being knowing that you're like very naive about their science is the number one thing I recommend. But also the space really needs more software engineers. So dive in, and with that mentality, and there's a lot to be done.
That's a really great answer. Thanks, Alfredo. So I'd like now to maybe take a step back and look a little bit at what are the main trends that you guys see. And this is not a question for anyone in particular on the panel, so whoever wants to take take this one, feel free to go ahead. But the first thing that comes to mind, of course, there's a lot of talk in terms of AI and what's the impact in biotech in general, but particularly maybe lab automation. And I want to ask you guys, have you seen that impact realized yet? And if so, we can talk a little bit more about that. If not, what's promising? Where do you think that impact is going to come in terms of AI in the lab? Anyone who wants to take this, feel free to go ahead.
Let me just think for a few seconds. I think the answer to your first question is I haven't really seen it. So I'm speaking about wet lab automation now. Haven't really seen it come into its own yet. I think people are still probing it out, especially if we're talking about like generative AI. I think there's a lot of piecemeal things that it could help with, like changing between formats. If the vendors would be up for integrating something like a simple chat interface into their tools, I think that could be immensely helpful to do some of the annoying things, right? I doubt that's gonna happen, but I think that could be useful. Yeah.
Alfredo, I'm curious to get your take.
Yeah. Well, yeah, sure. I'll go first, and then I'm curious on your take. But because my take kind of includes what you're doing, and I think that the most important things for AI, like, okay, so I'll give you a fun and slightly sexy answer first. Like, there's a lot of really cool ways to do AI in bio. Like for example, my co-founder is writing a blog post right now and kind of fine-tuning Evo, the new ARC Institute model, on like to generate like with a Church Labs data set to predict new AAVs. And then obviously there's AlphaFold 2 validation. We have like a lot of customers running thousands of proteins at once with small mutations and then trying to see the differences between them. And there's more and more and more use cases. But the reality is like the unsexy part of that, of AI, and the most valuable part of AI, what I believe is going to be the Briefly bios of the world. And you don't see AI anywhere in their name or almost anywhere in their homepage, but like synthesizing that information and making it very easy for it to be machine readable and very easy to be synthesizable by a machine learning model in the future. Vial's work doing the same thing for clinical trials and just like making it collaborative so that there's no kind of data silos everywhere, and automating a lot of the processes so that people are really like recording all of the data and centralizing databases and file storage. I mean, that's something that the three of us in this panel are doing. And by doing that, the reality is the other sexy things I mentioned and everything you hear in AI today, that becomes pretty simple. Like honestly, like as a field, bio is not really automating that much in AI. It's really taking things that are being invented in the AI space where there's way more data and way better organization, and then applying them to DNA, applying them to proteins. And the most impactful thing you can do today is help the field have better information, better recorded, better base layers. And then it's like a Maslow's hierarchy, but of data rather than humanity. It's like if you like, AI is really the very tip, and then everything below that is just like the basis of having, I mean, what I already mentioned. And if you have all that, then the very tip kind of comes much easier. Yeah.
I think that's totally spot on. I think we're missing a lot of base layers. We talk a lot about our problem is a problem of unstructured data becoming structured, and then paper becoming digital. So structured, digitize, automate. Everything is through the lens of structured, digitize, automate. And lab, preclinical, clinical, we have so much work left to do just to get the data structured, to get the data digitized, to get APIs that talk to each other. And only then, maybe at some point you can say, hey, write me a workflow automation that takes this data from here and automatically, and then we could have some great AI-powered automations. But we are, I think, quite a distance away from those workflows really taking off. Not because they're not powerful, but because we have so much work just to build the pipes, so much work to structure, digitize, and automate pretty much all the workflows. It's not just a healthcare story, right? If you go into old world Fortune 500 companies, the same thing is happening. I catch up with friends who are doing enterprise SaaS deployments into Catalog, into all these large companies, and they're dealing with the same problem. Their ERPs don't talk into their CRMs don't talk to, and so I think a lot of old industries are going through the exact same thing that life sciences is going through. And yeah, Maslow pyramid hierarchy of needs. We'll get there. It's just a lot of work to get there.
Yeah, totally. I mean, I agree, and I think we're still, as an industry, when you see this new technology come, it used to be machine learning, you know, 5 years ago. Now it's generative. And we just so want to apply it to the garbage data we have and hope that it's gonna produce all those beautiful results we have, right? And you know, we just got to start from the bottom up.
Beautiful. Thank you, guys. And then similarly, another topic that I think is coming up a little bit more, especially I think in relation to lab automation, is to some extent the rise of open source software in particular, I guess. And I'm curious, again, have you seen this really happen? What are some tools that you may recommend or you think are particularly useful in terms of open source? And do you see that as something that is actually picking up or not really in the lab automation biotech sphere?
Yeah. So I think we had like on the hardware side to begin with, we had the OpenTrons come out, OpenTrons 1 quite a while ago now. And I think that was fun. Then the OpenTrons 2 came, which was the promise of now we have an actual real device here that can compete with these other ones, but at an order of magnitude less cost. And as much as I love OpenTrons, I think it didn't quite come to fruition. I think a lot of people, at least in industry, felt like you can't take the risk on you know, saving money on that hardware investment if it doesn't quite, you know, do everything you want it to do. And so I think it's good that there's initiatives in open source, and I think on the software side they did an amazing job. They showed us what's possible, that we could be writing Python code, programming our liquid handlers that way. And I think that will take hold. I know there are other initiatives out there, and I think that I hope that their new platform is gonna be able to really compete with the big ones that are more expensive. But yeah, I'm positive about it, but I still haven't really seen the industry being convinced. Not in the same way where it has in software, right? Where open source is like it's a huge thing and it's used in every serious company. That's my take.
Yeah, Simon.
We use Documento, open source DocuSign. We actually just contributed to the open source community to make it CFR Part 11 compliant, the FDA requirement for software. So we're thrilled for open source. That's a fairly, I guess, to some extent, niche implementation of open source. I would love to hear more about Alfredo's takes on the open source lab automation community. And there's a lot happening there. We've played around with the OT Flex and kept up with OT. People seem to love it. I think there was definitely a turning point in the last couple of years where it's gone from hobbyist academic community to now more widespread. I was at SLAS a few months ago and people love it. There's a lot of enthusiasm for not only it, but what it means for the platform interoperability, building plate-based fluorescent readers that live on top of the OT that are tightly integrated by building open source. There's the whole movement to do the SILA 2 standards or this open source interoperability across lab automation, seeing a lot of new up and coming players build with SILA 2 in mind from the ground up. That's really cool. That's the type of things that we would have loved to see have happened in the SaaS community, open source, web edge community a long time ago. It's happening almost from the ground up in lab automation. So I think a lot of green sprouts to look at and get enthusiastic and excited about for the future of open source.
Beautiful. Alfredo, anything to add?
Yeah, I'll actually add something because, by the way, Simon, that's incredible that you guys added the CF compliance to that tool, and I didn't even know that existed. So definitely we'll look into that because DocuSign. Yeah, I mean I won't comment on DocuSign, but yeah, everyone knows. But I will say, like, I respect open source so much, just because I respect but it's in a type of respect that I have for like nonprofit foundations and the kind of foundations that are donating money to cure diseases and the kind of foundations that are giving money for water to give water to the world. I am much more pragmatic when it comes to building a sustainable business model, and there are a lot of dangers and perils in both sides, both in the sense of kind of building a true business model, like having open source traction by no means means that you can build a business on top of that. I think we've seen that with Docker. I mean, how much value has Docker added to the world? And as a company, they've regrettably not been able to capture a lot of that. How much value has Elasticsearch like added to the world? And as a, like, again, same story. Now there's also incredible success stories. I mean, you have Streamlit, which sold to Snowflake for $800 million. You have Terraform, which became HashiCorp and is now like an incredible companies. And so there are definitely ways, but it is by no means a business model. And sometimes I would like trade off some companies, do a little more closed source, but then they provide me that service for the next 10 to 20 years. Then like they like have to go out of business or they can no longer keep running the thing. And the other thing is like, a lot of people think like, oh, you put it out to the world and then Simon comes and then he contributes this incredible thing. And there are like really kind people like Simon in the world, but they're very few and far between. And the reality is that a lot of the times, like maintaining a project open source and having the quality and the bar and the carefulness that you need when something is open source is actually ends up being much more work than the work you get from the community. And it's a very typical myth that like, oh, you'll have people contribute. Like most projects don't have anything to contribute. That said, with all that said, I think open source is one of the greatest, like, yeah, one of the greatest things to do for the world. And it advances the world in so many incredible ways. In bioinformatics itself, all of the bioinformatics workflows that people contribute are amazing to the world. And so, yeah, huge fan, still pragmatic, but yeah.
Absolutely. Thank you guys for giving your 2 cents. I have one last question on my end, which I would like to be a quick fire one, and then we can open up some questions from the chat, which I know there's already quite a few. So on my end, I'd just love to hear quickly what is that you guys are excited about in biotech in general, and then lab automation and CRO, if there's anything in particular, and anything that comes to mind that you guys are excited to see coming, and maybe the audience can go and pick it up as well. Anything that comes to mind?
Yeah, one, selfishly, we're about to kick off the clinical trial for our first internally developed program, which is a huge accomplishment for our team. So we're personally very excited to be now taking the step of being a clinical stage biotech company as well as a clinical stage CRO for other companies. So very excited by that. We'll be talking about it a lot more soon. Keep up with our story. And in terms of the broader community, I think we're going through a generational shift. I think the biotech community, it looks a lot more like technology founders did at the beginning of the software revolution. And that's remarkably exciting. I'm meeting a lot more founders who come at things from a first principles mindset, who are looking to change the dogma that biotech and life sciences comes from. I think credit to the panelists here on the call. There's just a lot of I think talent and momentum coming into the space. And if you project out where that is going to be, if not only just a few years, but further out, we're going to be in a completely different place. The industry is going to be completely different. So I'm really excited by what we're going through.
Yeah, I'll tag on to you there. I think seeing the field of engineering and field of science kind of merging together in this new wave of biotech is really exciting, and I think it's gonna potentially speed up the way we do science by a lot. So that's super exciting. And then on a less visionary scale, I think I'm very excited about a few more acoustic liquid handlers for people to buy at lower cost than the Echo. And I saw that at SLAS. Not going to plug in anyone, but I think that's cool and that's needed. Can't be paying that many hundreds of thousands to get one of those, but they're very useful. So yeah.
Alfredo.
Yeah. I'll go a bit more kind of visionary and futuristic for this one, since I've been like quite pragmatic for the whole panel. But I think I've told this story to Simon, but I was visiting an old lab the other day. I built as recently as like 10 years ago, not that old, maybe like 2010, and they had kind of some cell counters in there and some like 6 well plates, 96 well plates, like typical lab that you'd imagine, right? And then I was visiting Ginkgo's facility, specifically the one where they were doing thousands of COVID tests per day. It was completely automated end to end. It was like my first time seeing a 1,536 well plate, like 50, it was the same size, just like these beautiful, like little tiny wells that you could not possibly imagine anyone pipetting by hand, right? And they don't. They have they don't have a single biologist doing experiments by hand. They were mostly in that facility. They were mostly like operating the machines and then like moving them from like one machine to the next, like the plates themselves. But all the liquid handling was done automatically. Now, if we kind of take that a step further, and those like you take those away, those last few humans, and then you make everything APIs that control these machines, and we're far away. Again, it's a dream, but you take out those last humans, and then you get kind of like what Stratios is doing, but like futuristic and like a true cloud lab that you can use, like a future where all biology is defined and executed through programming, right? Like a Python defined protocol, like all the data analysis is being done automatically, seamlessly. And so it's just all one loop that can be like a microprocessor done thousands and thousands of times. And so that combined with other innovations will bring down the cost of biology by many, many orders of magnitude, like it has done for computing. And the final dream is like in software, if it was 1990s, you had to ask investors for a few $1 million for a Series A. You would go and buy servers and then build for many months, and just to get a software business off the ground. 10 years ago, 10 years later, like around 2004, Mark started Facebook from his Harvard dorm room with a couple $100. That is incredible. And that is the future that I dream for, where a college kid has an idea for a new therapeutic modality. She spends $1,000 to send off the experiment to a cloud lab where the Python script gets executed. And by the time she wakes up, an ML model has iterated on it 100 times, and maybe it shows her that nothing is there. But maybe if that kid happens to strike luck, like Mark did, he just found a new drug modality and she's off to the races to put it into computational mouse models to test for toxology. And then if it all goes well, send it off to Simon to do the automated clinical trials and get a drug to therapy 10 or 100 or 1,000 times faster. So that's kind of the future I'm excited about, and we're far away, but I think it's a very exciting one worth building towards.
Very inspiring, everyone. Thank you, guys. I'm going to start going through then the questions from the chat. There's already quite a few. Anyone else who wants to add some more, feel free to do so. And the first one is for Simon in particular, and is how did you learn the ins and outs of CRO's operations in the first place? And then secondly, in the whole process of planning and conducting clinical trial, where do you see the real potential for using LLMs?
Yeah, first question, my co-founder and I didn't come from life sciences. We didn't study it. We knew nothing about it. We came from real estate and commercial insurance and fintech broadly. And I would give new first-time founders the advice of just get into the workflow, use your interviews, clear your calendar all day long, talk to people and talk to them about their pain points. Don't talk to them about technology. Don't talk to them about market sizes. Don't talk to them about what they hear competitors are doing. They don't know anything that they just know their pain point. They know what they deal with on a day-to-day basis, and just start from that. Start from the pain point. You go do the work, map the market. You go do the work, understand the integrations, the software competitors, how you want to enter the market, the wedge, but just really deeply understand people's pain. And so that's exactly what we did. We've done thousands of user interviews through the history of the company, and at every different point in the user interviews, we're very focused. So let's go deeply understand the clinical workflow at the clinic. How do you capture user data at a visit? Let's go deeply understand the data management process. How's data captured, structured into the CDASC format for the FDA. It's good to deeply understand. And you just, over the course of tens of user interviews, you can come to a pretty strong understanding of how things work. So basic product approach, but just I would be very, very deliberate, but focus on pain points. LLMs, a lot is happening here. A lot of people are trying protocol generation via LLMs. A lot of people are trying safety monitoring reports via LLM, a lot of other things. I think the real question becomes how much can be done via like templating and automation of just kind of your basic process automation versus what really needs to be truly LLM. There are definitely cases of kind of overdone medical writing and things that are administratively burdensome that are done manually that probably can be made more efficient by an LLM. But I think that I think the question is maybe written with a mindset of is this a company? Should I start a YC company focused on protocol generation via LLM? Or and I see a lot of those types of companies. I would say from my vantage point that there isn't, I think, room for that. It'll be part of a broader platform. It's a fairly niche implementation, and largely structuring, digitizing, and automating is what we need more. There's a lot too much of what happens in the administration of the clinical trial, of writing the protocol, safety reports, IB writing, etc. That's where commenting back and forth, stuck in the morass. That needs to be structured, digitized, and automated. And then later on, maybe some small workflow automations via LLMs, but it's a very small part of the value creation.
Beautiful. That's very insightful. Thank you, Simon. The next question is just for the panel in general. So whoever wants to take it, feel free to do so. Can you describe how you acquired your first customers? Were they former partners, or did you just start the relationship as buyer-seller? And then did you leverage any partnerships at all when building the tech? So that's a sort of separate one. How did you go about building the tech? Did you leverage any partnerships? And what did you think helped you succeed in securing that partnership and pilot? So first one is around acquiring customers, whether they're partners or just like starting from scratch.
Yeah, I'll just add a quick, I guess a plug there is the Century of Biology has this great blog post on Vile and how Simon went about doing the work for the discovery and just everything he talked through. And I think it really shows kind of the obsession that you need and just all the work that needs to happen before you build like your first line of code or before you build your first line of product. And so definitely go and read that. And just yeah, +1 to that answer. It just like a lot of people ask like who should be your first like customer, like how do you find your first customer? It's like no, like that's like the wrong order. You first need to go and talk to users, and then they tell you their problems, and then you get enough users, and enough might be 3, it might be 6 that have the same pain point that you want to go solve. And it's like, how do you get the first customer? Well, ideally they're the first customer because you've already asked them, will you pay for this if I solve it? And they're like, yes. And they're not just like, yes, they're like so excited, like so hair on fire problem for them that they will literally be like, yes, I will pay you the moment you have a solution for this. And again, yeah, Vile's blog post does a great job of outlining a framework for doing this. But in general, like it has to be a very pragmatic, and this kind of touches on LLMs too, right? It's like no, like go ask them about their pain points, and maybe like LLMs are the best way to solve that. Maybe it's sticky notes, and like you'll figure that out later. But just kind of focus on those pain points.
Beautiful.
Yeah, I mean, I think that's true, right? Like during the early stages, when you're figuring out what problem to actually solve, you talk to so many people. So you build up, if you don't have a network before, you will build up a network then, and that can be your first sphere of people that you would approach with you know, an actual solution. And like the other guys say, like hopefully then there's someone who you already know has the problem, and so they'll be your first go-tos, I think.
Beautiful. And in terms of the second part of this question around leveraging partnerships to actually build out your tech, did you guys do that at any point? And if so, what helped you succeed in securing that partnership or pilot?
We're doing that right now, so I'm not gonna. But I mean, I think it's the same kind of answer for us. Like that comes from those people who are interested. It's just like the next step for them, is what I'll say.
Beautiful. Next question is.
Oh,
yep. Sorry, Alfredo, you were saying.
Oh, sorry. I'll just add to that because kind of Stefan was like just they're getting started, and I think they've clearly done that really well. The product seems to be pretty beautiful, but it's just in general, I would say like that never ends. That is day 1, and that is like day 1,000, and that's day 10,000. It's just like we have 60+ biopharma companies today using Latch, and they are each our partners. And if we're not meeting with them once every 2 weeks max, and usually with our best ones every week, then we're not doing our job correctly, and we're not adding enough value, and we're not iterating quickly enough. And so to this day, we meet with over 30-40 customers every 2 weeks, and we just ask them like, what can be better? What are your other pain points? What are your adjacent pain points that we can be solving? And that's how you start day 1. You just like ask them about a single pain point, and you start expanding from that. But you got like that obsession is the most important thing. And if you do that well, then you're building long-term partnerships that will like vastly pay off.
Beautiful. Thank you, guys, for the answer, both of you. And the next one is again for everyone. So whoever wants to pick it, feel free to go ahead. Is about the particular hurdles that are inevitable at the start of an AI startup in clinical trial optimization, particularly. And so what are the hurdles there, and what skill set would you say is someone with a scientific or business background lacking that are essential for launching an automated software company for the pharmaceutical industry? So this is more about the skill set. Maybe we can first focus on the first one. What are the hurdles of starting an AI startup in clinical trial optimization? If anyone has anything to say, maybe Simon?
Yeah, sure. It's okay, maybe I'll take the skill set question. I think, yeah, I think the skill set I think there's a lot of life sciences problems that feel scary, right? The language is very difficult to learn from the outside, or perceived to be difficult to learn. And I think broadly, what we've certainly had the experience of is a team of sharp generalists who starts from a first principles mindset and comes at a problem, whether it's a lab automation problem or a how do I solve this really difficult operational problem and structure it as an automated, or how do I do data management via computational approaches at scale. All these problems can be solved, and I would generally push that if you come from a software background, if you come from having worked in biz ops or finance and strategy at an operationally intensive software company, you're very well suited to come into the space and have a lot of impact. In fact, I think you're kind of best suited to have come from. If you've seen how DoorDash or Uber, a lot of these other companies operate, that's the kind of approach that we need. We need process adherence and really tight integration between various tools, and we need metrics obsession in a way that I think a lot of life sciences companies don't have today. So I think the right skill set is one. I don't think it's important. I think any sufficiently team of sharp generalists can figure it out with enough technologists and operators on the team. And two things are scarier than they seem when you get into it. I would not be scared by the lexicon.
Anyone else that wants to add anything? Nope. Cool. Then I'll probably move on to the next one, which is again for the whole panel. What are some of the ethics that potentially can act as a barrier for AI automation in biotech? So the ethical side of it.
Well, I mean, I can start with a very practical and like on the ground barrier right now. And I think that is the fact that you're sending potentially sensitive data somewhere else. I think that's the that could be data related to patients, or it could be IP for the company. And I think sending that to another company, there's a lot of pharma companies, especially I guess in our industry, that wouldn't be too happy about that. And so then that becomes a stopper for people who want to use it. I can't think of any other ethical considerations off the top of my head. I don't know if the others have anything.
I'll just add. Oh yeah, yeah. I'm definitely nowhere near the equipped to kind of answer deep here, but I will say, I mean, I think biodefense is going to be more and more of an interesting space. We're gonna really need to invest in it. As a country, I think we will need a lot of investment going into biodefense as these models and just biology becomes better at just engineering biology. A lot of that comes with like negative. Every technology comes with negative side effect, possible negative side effects, and I think the best way to mitigate those is by inventing them, and then inventing them before everyone else, and then mitigating the side effects before they become something dangerous. And so I think that will become a very important field. We need more investment going towards that, and we need to make sure that the US maintains a leadership in that space throughout. Yeah.
Beautiful. And I'm curious also, like, if the ethical side of it that Staffan mentioned is also relevant for CROs, where it might imply sending some of your data to like an external organization that then does something with it. Do you see any any regulation in particular, or anything that came up in your experience, Simon, around that as well?
In many ways, there's almost two questions here. There's compliance and how do you build a highly compliant, scalable organization, and compliance takes the form of your software and your people. And I think everyone here builds highly compliant software and needs to maintain compliance with, at the very least, HIPAA, in many cases, also SOC 2, in many cases, ISO, and we deal with CFR Part 11 compliance. And that's critically important to build from the ground up. It's a lot of process adherence, and it's a lot of system design to make sure that as you're building releases, that everything is adequately checked against your core compliance system. So that's the first. The second is people. Really important to, I think, hire a quality team early, get your people systems around training. Everyone in our company is trained in either GCP or GMP, depending on what side of the organization they sit on, and they're constantly going through that training process. So I would think that the first question here is just that. On the true ethics side of it, we work with Australian regulators, and the IRB equivalent there is the HREC, which the E in HREC stands for ethics review. And it is critically important whenever you're running a clinical trial, you're getting a patient's consent to everything in the investigator's brochure and the protocol. And so the concept of ethics and consent and alignment with the patient is something that we think about from the ground up. Everything we do, from the e-consent process, which is the very first step in our clinical trial, where you review electronically the protocol and you consent into it. That's a user experience. That's how do I ensure that you're fully aware of what you're signing up for in entering the clinical trial. And so we think about it quite a bit. In fact, we think it's one of the places where the field today is doing a rather poor job, where you get sat down in a room today with a big stack of a protocol, and you're actually given 20, 30 minutes to read it. The protocol is likely not written for a grade 8 level of reading. The protocol doesn't have any clear highlights. It's not in video format. There's not bulleted form. It's very difficult to make sense of what you're actually signing up for. And what you end up seeing is a relatively low NPS and a high dropout rate. So there's lots of room for the tech community broadly, as they come into the space, to up-level how we bring patients into the life sciences community, especially when it relates to clinical trials.
Beautiful. So we'll have to close it here. If anyone has any more questions, I'm sure you can try and reach out to the guys on the panel. I'm sure they're happy to get back to you with any responses. But to be respectful for your time, which we appreciate a lot, guys, thanks for coming, Staffan, Simon, and Alfredo. We'll close it here. So thank you, guys, again. I appreciate it a lot. I think it's been very insightful, at least for me. I hope for everyone else as well. If there's any more topics that anyone in the audience is interested in, Nucleate are going to publish a resource page on CROs and automation, where you can find all the things that we mentioned on the call, plus some more resources as well. And then on top of that, in the chat, you can find a link tree, which brings you to all the relevant content that Nucleate produced and will produce as well in the future. So stay tuned for more events like this and more content as well. Again, thank you for our amazing panelists today, and thank you for your time. We appreciate it a lot. And I wish everyone a great rest of the day. Thank you, guys.
Thank you so much, Lorenzo.
Thank you.
Thank you so much, Lorenzo. Great to see you all. Bye.
Thanks, guys.